So before we get started on Lecture 10, DNA and chromosome structure, I just want to say a few words about the first midterm. Overall, I think it went very well, as you can see by this distribution. About 42% of you got a 90 or above, which is very good. If you got a grade that was lower than you would like, and please come see me about that. We can talk about different strategies for studying, talk about what might have gone wrong, and yeah, I can help you kind of change the way you're approaching this course for the next exam. The mean was, I believe, something like an 83%. So everybody did quite well. Not everybody, but on average, yes. Okay. All right, so this lecture is going to talk in more detail about the chemistry of DNA and DNA structure, as well as chromosome structure in more detail. And we'll also cover things like telomeres, centromeres, and mitochondrial DNA and mitochondrial chromosomes. So even before people knew or understand what the structure of DNA was, biologists, geneticists, recognized that the genetic material, whatever it was, needed to have a number of characteristics. And one of this was, first, it needs to be able to store large amounts of complex information, because people recognized that the entire phenotype of an organism was encoded in this genetic material that was passed down from generation to generation. That's a huge amount of information that needs to be stored. Second, the genetic material needs to be able to be accurately replicated and faithfully inherited from generation to generation organismally, as well as from cellular generation to generation through repeated cellular divisions. Third, the genetic material needs to be able to encode the phenotype in its entirety. And fourth, the genetic material needs to be variable to account for the variability that we see in phenotype. So naturally, DNA does all of these things. And I just want to take a moment to take a closer look at what exactly are the building blocks of DNA and what do they comprise of. So DNA, as you know, is comprised of nucleotides that are made of a base, a sugar, and a phosphate. And these are repeated throughout the DNA. So the sugar is consists of a carbon ring with an oxygen. The most important thing on here is that on DNA there is a hydrogen at this 2' carbon, whereas at RNA there's a hydroxyl group at this 2' carbon. So the sugar, the real difference between RNA and DNA is with the sugar. And so the ribose sugar consists of 2 hydroxyl groups of the 2' and 3' carbons, whereas deoxyribose only contains a single hydroxyl group of the 3' and a hydrogen in the 2'. The base is where the A, C's, G's, or T's comes in, or U in the case of RNA, and they come in 2 flavors. We can have purines, which are 2 rings, and these are adenine or guanine, or pyrimidines, which is a single ring structure, and that includes cytosine, thymine, and uracil. The phosphate group is just this structure, and this is where we get the overall negative charge of DNA. And the phosphate is what gives DNA its acidic nature, why it's a nucleic acid. All right, so let's take a closer look at the nucleotides. I forgot to mention, actually, I guess we can see it here. So these are nucleotides that are just the phosphate, the sugar, and the base, all together is a nucleotide. If it is just the sugar and the base without the phosphate, it's a nucleoside. So you may hear that term now and then. I just want you to understand the difference, that they're basically the same thing. One has the phosphate group and it's a nucleotide. If it does not have the phosphate group, it's a nucleoside. I also want you to understand that the phosphate group is always located on this, I just want to make sure I double check where exactly, whoops. Oh, there we go. Okay, so the phosphate group is always attached to this 5' carbon. And the OH group is always attached to the 3' carbon. So this is the 5' carbon. This is the 3' carbon. The phosphate group is always attached to the 5' carbon. This gives the directionality to the DNA. That gives it the 5', 3' directionality. Okay, so the phosphate group is always on the 5' end, and the OH group, the hydroxyl group, is always on the 3' end. And this is important because when a new phosphodiester bond is made between two nucleotides, it always happens such that the phosphate interacts with the hydroxyl to create that bond. And so if you have, so if you have a growing strand, let's say here's the end of our strand, here's our OH group, it's going to, the new phosphate is going to come up and interact with that. And so things are always added. That's why things are always added to the 3' end. Okay, so just to go back, so we have a deoxyadenosine 5' monophosphate, DAMP, DGMP, DTMP, and DCMP. And these are all the 4, and then you can have a diuracil also. Though, of course, that would be actually a different structure because you would have a ribose here instead of a deoxyribose for RNA. So these are the 4 DNA nucleotides that you can have. And then again, DNA, as we all know, is anti-parallel in nature, such that it's 5' to 3' on one strand, and then it's the opposite direction on the other strand. I also want to point out that GC bonds are comprised of 3 different bonds. AT bonds are 2 hydrogen bonds, and as a result, the GC pairing is stronger than an AT pairing. So if you have a GC-rich region, that means that that region of DNA is more tightly bound, and it requires more energy to dissociate or denature those 2 strands than if it were more AT-rich. Okay. So DNA structure has, just like in proteins, there's a primary, secondary, and tertiary structure. The primary structure of DNA is basically the order of the nucleotides and the order of the bases. And what we've been seeing in terms of these nucleotides and how they're structured, that's what we can think of as the primary structure. For the secondary structure can have 2 forms depending on whether the DNA is double-stranded or single-stranded. If it's double-stranded, the DNA secondary structure is the double helix. If it's single-stranded, I just want to say that double-stranded DNA tends to be, because it's supported by these hydrogen bonds, it's a much more stable structure, and it's actually so stable that we can get, quantifiable DNA and amplifiable DNA out of, very small quantities, and that's what makes forensics possible. Because DNA is a very, very stable structure, you can extract DNA from bones, that are thousands and tens of thousands of years old. And the nature of this is because it's double-stranded. So DNA that is single-stranded is much less stable because it has these open hydrogens that are looking to pair with something. And as a result, they often form these secondary structures that can form when you have regions of complementarity within a single sequence. And so it may loop in on itself and form a stem loop structure. Another form of this is just a stem without a loop. And keep in mind also that you don't necessarily need to have exact pairing going on in order to be able to form secondary structures if you're a single-stranded piece of DNA. RNA is usually single-stranded. There are some cases where RNA is not single-stranded, but usually in our bodies, RNA is single-stranded. And as a result, these types of structures are very common in RNA, and this is just one example. And they can get very complex, and they can also play a really important role in how the RNA functions. And so we'll learn more about RNA later, but RNA actually plays a huge role in all kinds of different processes within our cells and tissues, and a lot of their function is a result of these secondary structures. And so one theme for this section of the course, which is largely going to be about molecular genetics and how and molecular mechanisms for genetic processes like replication and transcription and translation. A lot of how these processes work relies heavily on secondary structures of especially RNA, but then also of DNA. And we'll talk about this in a few minutes, actually. Okay, so the tertiary structure of DNA has to do with how DNA is packaged into the cell. So as an example for how this problem is solved, we have a representation of a bacterial chromosome here, along with what would be to scale the size of the actual bacterium. And so a chromosome of this general size needs to fit inside this tiny, tiny space, which means that it needs to undergo tertiary structural changes that influence the way that it's folded and packaged within the cell. So the way this happens is in large part through supercoiling. And the way that you can think about it is if you imagine a coil, such as with an old-fashioned phone with a phone cord, you can imagine if it's over-rotated, it will supercoil in a certain direction, and if you under-rotate it, it will be supercoiled in a different direction. And so generally most DNA is negatively supercoiled. That is, it's under-rotated, and this is because the processes that require getting at single-stranded DNA, breaking strands, unwinding the helix, allowing proteins to access for transcription, etc., require that it requires less energy to do all of those things to the DNA if it's negatively supercoiled, that is, if it's under-rotated rather than if it were over-rotated. Oops. Okay. So there are proteins that are used both in E. coli and in eukaryotic cells, in prokaryotic and eukaryotic cells. There are proteins that hold loops of the DNA, and then these loops can be supercoiled for sort of more complex packaging. There are additional enzymes that help with the coiling and uncoiling of DNA, and these are topoisomerases. Some topoisomerases both coil and uncoil, but others only do one or the other. Okay, so in eukaryotes, eukaryotic DNA is structured around nucleosomes that are comprised of histones, and we'll see exactly what those look like in the next slide.